All right. Daniel, I guess this is your cue.
All right, awesome. Thank you so much. So my name is Daniel Wiebke. I am the president of the North Carolina Water Resource Association, NCWRA, and I'm just here to give a few announcements and then introduce our speaker for today. I appreciate everybody joining us for this pre-lunch luncheon, lunch and learn as we usually call them, but this is more of just a forum today. So with that, I'll make these announcements here. So an email went out last week about nominations for the NCWRA board. We're looking for volunteers to that would like to be nominated, or if you know somebody who has ties to the water resource profession, feel free to nominate those people. The link has been sent out. If you don't have the link, feel free to email myself or Frank Lopez. We can put our emails here in the chat later on, but those will be open till December 8th. And then subsequent to that, then we'll have elections. From there, we also are looking for volunteers for NCWRA. We have the membership committee, the program committee, and the communications committee. So you don't have to be a board member to be on those, but those are just slots and positions that help our board members who chair those committees bring the programs and the opportunities that we have. I know we've had a few less things happen the past year and a half, 2 years almost now due to COVID, but hoping next year we can get out and see more people again. And then the last thing is we are hosting the National AWRA Conference in summer of 2023. So we will be forming a ad hoc committee. So if you're interested in helping us put on the national conference in summer 2023, feel free to reach out to us. And with that, I will introduce Dr. Paerl here. So Dr. Paerl is an aquatic ecologist who works in coastal, estuarine, and freshwater ecosystems. His research addresses aquatic nutrient cycling and primary production, dynamics, environmental controls, and management of harmful algal blooms. The Paerl Lab also focuses on assessing effects of human and climatic alterations of water quality and sustainability to coastal and inland waters around the globe. So this specific presentation is more North Carolina, and it's going to talk, as you read in the description, about the overview of the macro trends in water quality in the Pamlico and Albemarle Sounds, and offer the context regarding the impacts of our rivers that flow from the Piedmont, so us here up in Raleigh and Durham, and Triad and those communities, and explore the ways in which the climate change has been driving those impacts. So I'm very interested to see how what we're doing up here is really affecting some of those coastal communities. And with that, I'll let Dr. Paerl, take it away.
Okay, I'm going to share the screen here. Can everyone see that?
Yes, indeed.
Okay, great. Well, thank you first of all for inviting me. I think actually this is very timely because we've got some new information to convey to you, particularly from work we've started doing in the Albemarle part of the system, which I'll be talking about. But first of all, I want to stress my colleagues, starting with Nathan Hall here at the Institute, and lots of other colleagues, graduates, undergraduate students. We're currently right in the midst of a undergraduate semester at the Sea here at the Institute. And then lastly, citizen volunteers who have really been incredibly instrumental in helping us collect data and at timely intervals. So that's helped us do a better job in connecting drivers with responses in these systems. So let's see here. I'll click on this. Okay, so just a quick review here. Many of you are familiar with this, but we'll just go over it really quickly. I just want to stress the importance of the Albemarle Pamlico Sound system. It is the second largest estuary in the country. Actually, it's the largest if you count the actual seashore of the system. We're actually much bigger than Chesapeake Bay, although that doesn't go for funding, but it does go for waterfront of the system. It is the most important U.S. Southeast fisheries nursery, so lots of fish that are caught offshore commercially and recreationally spend some part of their time in the system. It drains a lot of North Carolina and southeastern Virginia through the rivers that you see there. And of course, it's had more than 50 years of ag and urban expansion, both in the upper watershed, but also in the coastal region, which has been accompanied by more nutrient loading. And we'll talk a bit about that more. I want to stress also the fact that it's a lagoonal system. And lagoonal estuaries have a long residence time because the water exchange with the coastal ocean is really confined to these narrow inlets that you see. And in fact, most of the water leaves by evaporation from the system. So what gets in stays in the system for quite a long time. And that is one of the reasons why it's sensitive to eutrophication from nutrient inputs. The last thing I want to mention is, of course, the fact that we're at the doorstep of the Gulf Stream and a lot of storms that come in from that direction, including 34 tropical cyclones in the last 25 years. Barbara and I keep telling each other, okay, we're going to leave now after this one, but we've been here for 40 something years, and I guess that's kind of an old story now. And then, of course, accompanied by flooding, and that flood water brings lots of nutrients and other contaminants to the system. We have been monitoring, as well as others, of the tributaries coming into the Albemarle Pamlico Sound. And there are, of course, signs of trouble that have to do with excess nutrient loading and other contaminants, leading to harmful algal blooms, exacerbation of low oxygen events. You know, the system is prone to low oxygen, but we're exacerbating that phenomena. And then, of course, fish kills and shellfish kills. And lastly, loss of SAV habitats. And I'll spend some time at the end of my talk about that last topic, which has really come to the forefront with the coastal habitat plan and everything else. Okay, and then lastly, of course, we're impacted by these major storms, which are increasing in frequency and rainfall. We've been working on the Chowan. My first project actually on the Chowan was funded by the Water Resources Research Institute back in 1980, I think, to help solve some of the algal bloom problems on the Chowan, of course, the northernmost river coming into the Albemarle Pamlico Sound. We worked on helping the state come up with a nutrient management strategy, which worked quite well. It was both a nitrogen and phosphorus reduction strategy. But then about 10 years ago or so, the blooms returned. And this is what we're seeing now in the system, just to give you kind of a pictorial essay of the blooms that we've been experiencing since, the mid-2000s on the system. And we're currently working on it to try to identify causative effects and what we can do about this. One thing we know, and I want to stress this, that Albemarle Sound, of course, is a really important water resource that we have. It's the main crab fishing region of North Carolina, also other fishing and recreational activities. It is a system that's showing a very clear signal of eutrophication. So what I'm showing you here in the upper left-hand corner is the TKN, total killed nitrogen, which you can see is increasing and has been increasing since the early 2000s. And then if you look at the graph here on the right, that's been accompanied by an increase in chlorophyll. So, you know, it's pretty clear that the system is becoming more enriched with nitrogen, and that seems to be linked to increases in chlorophyll. and increases in algal blooms. But overall, I think we're seeing the system going in the wrong direction, the Albemarle Pamlico Sound system. I know there's a lot of scatter in this data, and that's because we collect a lot of data on the Neuse. But chlorophyll has been increasing in the Neuse over time, despite the fact that we have a TMDL for nitrogen. And of course, this would be much worse if we didn't have a TMDL for nitrogen. So, you know, I'm looking at this in a kind of optimistic way. And then Pamlico Sound on the right, this is sort of a mid Pamlico Sound station that we sample as part of the ModMon program. It is showing a very, very, very subtle increase as well in chlorophyll, and that's not too surprising, being a big system and having the signal not be as clear as it is in other parts of the Albemarle Pamlico Sound system. But overall, you know, we're heading in the wrong direction, and that is linked to nutrients. And the next few slides are going to show you what we've been looking at in terms of which nutrients are causing the problem or enhancing the problem of eutrophication. And this is just our bioassay setup at the Institute of Marine Sciences. So we collect water from our riverine stations or any other stations. It is then dispensed in these cubitaners, and I think you all know what they are. They're flexible plastic containers that are really good for doing these assays. And then we compare treatments versus controls in terms of nutrient additions. And you know, the good thing about these assays is that they are easy to do. They are incubated under natural light and temperature conditions, and we can get lots of help. People are very enthusiastic about helping with these bioassays. So if we look at one from the Chowan, this is from one of the students from one of our classes that we run in the fall, Felix Evans, but she ran a bioassay that really was good. You can see the replication's excellent. It shows that the system, if you look, the C's down there are controlled, the N's are nitrogen, phosphorus, and then N and P together. And those are just triplicate treatment samples. And you can see that nitrogen as nitrate stimulates, phosphorus not individually, but if you add nitrogen and then add phosphorus, you get more than nitrogen alone. So, you know, we clearly nitrogen is the major driver in the system, at least from this particular bioassay. And the pie graph on the right just shows you the composition of the algae in that bioassay. And you can see that blue-green algae or cyanobacteria dominate the system, not too surprising. And green algae come in second, and then diatoms and other more desirable algae come in third. So, you know, we've had a shift towards, you know, undesirable phytoplankton going along with this nitrogen stimulation. This is a more recent bioassay that was run by one of my graduate students, Malcolm Bernard, and the class down at the bridge there at 17. So this would be right in between where the Chowan comes into Albemarle Sound. And you can see nitrogen as nitrate, ammonium, or urea. Those are all equimolar additions. All stimulate. Phosphorus not, and N and P together actually don't give you much more than N alone. And as you get more into the salt water component of our systems, phosphorus does not stimulate. It's only nitrogen is the name of the game. And of course, that's because those systems have been trapping phosphorus for a long time because they are marine or partially marine. And phosphorus cannot escape the system. There are no gaseous forms. So, you know, there's a lot of phosphorus cycling around in the system already. A lot of it is natural. So it turns out that nitrogen is really the key nutrient that dictates the productivity response. This is work from one of my graduate students and myself that we did back in the early 2000s, Liz Calandrino. She looked at the sounds, and that's the location there in Currituck Sound that she did the bioassay on. And you can see in nitrogen again, no phosphorus effect, N and P give you the same as N. So it's kind of a broken record there as you get into the more saline system. And then, you know, the issue is this continuing sort of enrichment problem that we're experiencing. Different forms of nitrogen may actually give us different responses in terms of the algal community. We're currently investigating that. So, it's kind of like this question of, is nitrogen nitrogen, depending on what form of nitrogen it is. And we'll get back to this in a second, because organic nitrogen is becoming a bigger part of the story as well. And the picture on the right is a drone picture that was recorded by Will Reckling, who worked with us from NC State. And you can see the line there that's released by the drone. We're now using drones to sample as well as image the blooms below. This is from some work that Dan Obenauer and his student have done, trying to sum up sort of the sources of nutrients that we're seeing in our coastal systems. And you can see that, of course, non-point sources tend to dominate, which is the row crop, CAFO, wild being the natural inputs, things like forest, stuff like that. It varies in different places, but you can see overall the non-point sources clearly dominate. The NUS, which we know the most about, point sources really only account for about 10% now of the inputs. And that's largely because the point sources have been controlled. And in all the instances for the wastewater treatment plants, they're now in advanced wastewater treatment. And you can see the results of that showing up quite nicely. So, non-point source, that's the issue and continues to be the issue at this point. And that row crop part of the pie, of course, is fertilizer, mainly fertilizer input that we're seeing in terms of nitrogen input. But CAFOs, which would include the animal operations, also contribute a significant portion of the nitrogen input. Okay, so we got a nitrogen limited system out there, and nitrogen is the name of the game. We recognized that in the news quite a long time ago, and of course the TMDL is set for nitrogen reductions to go down to 30% decrease over what we saw in the 90s. We haven't met that, and in large part that's because even though we have seen a reduction in nitrate, which on the bottom graph you can see are the red dots, organic nitrogen has gone up. So the net effect is we're not there in terms of the 30% reduction. If we were to just look at inorganic nitrogen, we're doing a pretty good job actually and improving our act there, but not so much for the total when you include the TKM. And this is data, by the way, from the Chowan Basin. That's in their recent report that was published last year. We think that this increase in organic loading, and TKN, of course, is total Kjeldahl nitrogen, which would be organic nitrogen and ammonium. That has been increasing since the mid-90s, and you can see pretty clearly the TKN going up, the nitrate tending to go down. But the really problematic thing there is that if you look at chlorophyll A, that's been increasing as well. So we think that this organic nitrogen is probably at least partially reactive. And, this requires further investigation. And we're looking at that right now with several projects that are looking at this organic nitrogen load. Where is it coming from? What's its bioreactivity? We're looking at some specific sources like leachate from land-based waste disposal sites. We just got a grant funded through the Attorney General's funding source with folks that are looking at leachate from landfills. But of course, there are other sources of organic nitrogen as well. And we're working with Chris Osborne and his folks at NC State, linking their ability to separate some of these organic nitrogen sources by fluorescence signal, and then seeing what its fate is and how it's used by the phytoplankton community. This is still really work in progress, but I think we're clearly honing in on this TKN issue. And it's not only an issue in North Carolina. If you look at it globally, organic nitrogen loading is increasing to many coastal waters. We don't really have a clear picture as to what's driving that, although later on in my talk, we'll talk a bit about storms and mobilization of upstream leachates and things like that. So it could be linked to an increase in more extreme hydrologic events that are causing more release of organic nitrogen from sometimes natural sources, but also, of course, urban runoff, stormwater runoff, septic systems, ag, etc. Okay, now I'm going to switch a little bit and talk a bit about the climate change issue that's also impacting our coastal zone. And I don't think I need to convince you that things are getting warmer, but if you don't believe that, you can look at the North Atlantic anomaly graph there on the lower right, which shows this average temperature increasing in our ocean waters. And then, of course, the graph there on the left in the center, which shows the increase in tropical cyclones that are impacting us. So two things are really happening that we think are relevant to the eutrophication issue: warming and more extreme events, which can lead to more nutrient inputs, obviously, through flushing. But then if we have a major storm event that's then followed by a drought, then you have kind of the perfect storm scenario, because you got the nutrients that have been loaded into the system, and then the drought will increase the residence time. And that's really the formula for establishing blooms in these systems. We think that harmful algal bloom taxa are increasing because of this climate change issue. In particular, cyanobacteria or blue-green algae are increasing not only in our waters, but globally, again, in response to these extreme rainfall events, but also increases in temperatures because they like it hot. And the hotter it gets, the better they do in terms of competing with other algae in the system. So there probably is a climate change signal that is included in what we're seeing with the nutrient enrichment as well, sort of a synergy of those impacts. The hydrologic alterations that we're seeing as a result of these storms exacerbate non-point source pollution. So that part of the pie that's non-point source may be increasing just because of more extreme storm events and release of nutrients and other contaminants from our watershed. And the really troubling signal is that we're seeing more wet storm events over time. And this is from a paper that we published in Nature Scientific Reports back in only 2 years ago. And what we did was we got the data from the NOAA repository in Florida, the Hurricane Center. And there's actually very good data for North Carolina coastal waters because the mariners and other coastal interests kept good tabs on storms, the wind, and most importantly, how much rain fell from those storms. And what you can see from this data is that based on data that goes back to 1898, I believe, or somewhere in there, up until 2020, 6 out of 7 of the wettest storms that have been recorded in that data set have occurred in the last 20 years. So this is not a fluke, this is real. And what we're seeing is more rainfall, in part due to warming of the ocean water, picking up more water vapor. That water vapor then translates into rain as the system is bearing down on our coast. And then the other thing that's happening is that these storms are getting stalled more often. And I don't need to remind anyone of Florence, for example. But Floyd back in 1999 was another one of those storms that it was not I think it was a category 2 when it actually made landfall. So not a really windy event, although that's plenty of wind for us, but a very rainy event. And we're seeing more and more of these events over time. So that forespells more flooding, of course, and more discharge into our coastal systems. And that's what you can see there on the left-hand side. I'm going to take you now through this a little bit to go back to 99 when we had Floyd, but also discuss this in a kind of a bigger context and talk to you about what this means in terms of nutrient input and organics, the input of organic matter. So one question is, how much are these storms affecting loading of nitrogen and phosphorus in our coastal systems? And this is data from MODMON, which has, of course, been out there collecting data since 1994. Actually, it originally was a WRI project that then got funded through the Floyd Emergency Relief Fund. And then since then, we've had funding from legislative sources. And we just found out that, thank goodness, MODMON has been refunded for 2 years. Thank you, legislature. We hope to see the money soon. But anyway, if we look at years when we had storms, and let's go over here to 96, for example, you can see that most of the nitrogen loading is actually due to these storm events, even in relation to the spring runoff, which I would sort of call the natural input of nitrogen, although a lot of that is still really tied up in non-point sources. But then in years when we don't have a storm, you can see there is, it's pretty much dominated by the spring runoff. Here's 99 with the 3 major storms that we had there. Here's Matthew, Florence. Clearly, in years where we have storms, the loading is can be dominated actually by the storm events themselves, the total loading. And this is just the influence of these storms on material loads in the Neuse River, the percent increase over baseline due to storms, TN, clearly TP, but also the soluble forms of these nutrients increase quite a bit above baseline. So, you know, storm events are a big source of the loading, both for N and P in these systems. If we then link that to the algal growth response that we see in the Neuse, this again is MODMON data, and I picked out 4 events that we have good data for. And what you can see there are spikes in chlorophyll, the algal growth response parameter to these storms. There are other peaks that you see that are not necessarily tropical storm events, but they are extensive rainfall events, particularly in the summer period. So here's some. So it isn't all about hurricanes and tropical cyclones, but they are having a pretty measured impact on our total nutrient loads. And this is a summary graph. It's pretty complicated, so you know, kind of look at this in a kind of a broad sense. What we show here is the discharge at New Bern, freshwater discharge at New Bern into the Neuse, and then the chlorophyll response. So the redder it gets, the darker green and redder it gets, the more chlorophyll. And you can see with certain storm events like Ernesto here back in 2006, the major storm events in 1999, 2000. And then more recently, Joaquin, which was a double whammy kind of storm event that sat offshore but delivered a lot of rain. You can see there's a big response there. And then more recently, Florence and Dorian and Matthew as well. So there is a pretty good relationship between these extreme events and the phytoplankton growth response and bloom activity that we're able to capture. If we go back to 99, with the 3 major storms that hit, of course, Dennis, Floyd, and Irene, they all hit within 6 weeks, and we were ready to pack up and go somewhere else after that terrible set of events. It was pretty awful here in Beaufort. These were Category 3s before they made landfall. They occurred within 6 weeks. Record rainfalls in the Neuse Basin, many places exceeding the 100-year interval. And then we had some 50 to 5-year flood events in the watershed itself. The bottom line is that Pamlico Sound received its annual input of nitrogen in 1.5 months. And you know, when we go back to that other graph that shows you the big bump there, you can see that it probably dominated nitrogen loading for the entire year. This is the before and after picture, just to give you an idea of how large this impact is. This is from CWIFS, the satellite that's no longer up there, but it's been replaced by MODIS. So it's a real, color image before here and after. And the thing I want to point out here is this dark brown bolus of fresh water, of course, with sediments and organic matter in it coming in after the storm event. And this really gives you a good feeling or impression of the lagoonal nature of Pamlico Sound. It's sort of like a giant bathtub, it fills up. And then if you get an extreme amount of freshwater input, it'll overflow. And here you can see the sediment-laden waters being taken out to the Gulf Stream offshore. But this, I think, gives you a good appreciation for the loading that we can get in the system, and also that a lot of this stuff stays in the system for quite a bit of time, so that you can get a lot of regeneration of nutrients and organic matter with follow-up blooms. And that's what we see in this next slide. So here's the 3 storms that are hitting. This is one of our sampling points out here in the middle of Pamlico Sound, or in the southern part of Pamlico Sound. And this is the normal sort of salinity that you see out there. It varies between 15 and 20. Then we get this big freshet from the storms, which essentially drove the system, made the system fresh. And of course, that's a whole big stressor in and of itself in terms of fish and shellfish. But the really important thing is that it took about a year for the salinity to normalize again and get back to what it was before. So it ain't over when it's over, I think, is the kind of the take-home message here in terms of hydrology and salinity. If we look at chlorophyll, again, the growth response of algae, you can see clearly that there have been several spikes, not just only immediately afterwards, but some of them occurring actually in the following year. And that probably has to do with the residual nutrients that are in the system being used to support blooms in the following year. We'll get back to that in a bit. These blooms were largely dinoflagellate blooms, mostly harmless dinoflagellates, although there were some harmful ones that were categorized by Pat Tester and her folks at the NOAA lab, who we work closely with on monitoring the system. The other important thing to realize when we have hydrologic variability is that the action in terms of algal blooms changes from upstream when the flow is low. So this arrow is essentially low flow. When the flow is moderate, you're getting more freshwater input, but also more nutrients coming into the system. And this is coming down from the Albemarle, by the way, into Pamlico Sound. Then if we get a really high event like a tropical cyclone, the rivers start acting like pipelines, essentially. The residence time is too short in the rivers to develop algal blooms. So we see the blooms developing more out in the open sound system. And then here we go back to lower flow. And again, the impact is more in the river systems itself. We've been able to, using ModMon, we've been able to pick off specific bloom events and document them in terms of what was in that bloom and what the issues were in terms of environmental and human health issues. And in this particular situation with Eresto, which occurred in 2006, here's the freshet coming into the Neuse. So you're kind of looking at the Neuse sideways here, going into Pamlico Sound. Here's the salt water trying to make its way up into the Neuse. This, of course, is carrying a lot of nutrients and fresh water, and it sets up a salinity gradient in the system. And that has led to, so the residence time increases, as you move down. That allows the algae enough time to develop a bloom here right at that interface. And in this case, it was a bloom of a toxic dinoflagellate, Carlodinium. And then the various sources started to report fish kills that occurred at this place here, exactly in the same place where we had the bloom. So in this case, we were able to really document, the impacts on the bloom itself from the nutrients and freshwater input, but also the linkage to fish kills. in terms of the number of cells of carlidinium and the toxin that was produced. This data, by the way, came from our colleagues at NOAA in Beaufort, who did the toxin and cell analysis. So, there was a bloom that developed right in here, midway down the noose, and fish kills on both sides of the bloom. And they were due essentially to carlidinium, which kills fish by producing a mucus that clogs up the gills of the fish, and then they suffocate. So, this has been pretty good detective work if you combine MODMON with the activities that we can muster from our colleagues in Beaufort. So this is kind of the big picture then, the interactive effects of nutrient inputs, climate issues, including warming, which will lead to more stratification, stability in the water column that some of the bloom organisms like the cyanobacteria. And by the way, the cyanobacteria are really good at dominating once they get into the system, because they bloom at the surface and then they can shade out anything underneath them. So a lot of the more desirable plants and algae growing in the system are suffering because of this light shading during a bloom. And the bloom can move up and down in the water column. So at night they can get nutrients, and then they go up to the surface during daytime to use sunlight to grow. And again, the perfect storm scenario then is high flow, bringing in lots of nutrients, followed by a dry condition or low flow, allowing the bloom to form in situ in the system. One thing I wanted to also stress is that organic matter plays a very important role in all this. And this is data that we've been working on with Chris Osborne at NC State and his colleagues. Chris is a geochemist who has really got the expertise in terms of the analytical aspects of looking at organic matter in the system. We get this brownification that goes on with the increase in organics, and you can clearly see it here as stuff overflows and goes into the coastal ocean. If we look at the relative contribution of organic matter, this is dissolved organic matter, again through the MODMON program, you get a very similar picture to what you see for nitrogen. In years where we do get major storms, that DOC loading, and here you can see it, DOC and POC dominate the total annual load. So we're very much at the mercy of these big storm events that are increasingly impacting our system now. And this is kind of a graph that we put together to look at what happens when we have a big flush from an event. This is from Matthew, I believe. And here's discharge. So here the storm hits. This is the increase in discharge. That, of course, flushes out the algae because the residence time is reduced. But here's the organic matter input from that. But it didn't take long for the algae to come back and bloom afterwards. So, they're using, of course, the nutrients and organic matter that was input by the storm event earlier in the system. And again, keep in mind, except for the flushing events, these are long residence time systems. The noose, for example, the residence time can vary from 30 days to over 90 days. So that's plenty of time for blooms to form in response. I think I've already mentioned this, so we'll skip this one. Oh, the one thing I do want to point out is that transparency, of course, is reduced. And we're going to get into that now with regard to seagrasses. We know that seagrasses have been suffering in our system, and they're very much at the mercy of how much incident light strikes them. Seagrasses need at least 10% of the surface light at the bottom to grow. So if we look at over time what's been happening with our seagrasses, and this is data from Judd Kenworthy's work, by the way, that's continuing, doing AppNet work. You can see there's been a decrease in seagrasses even in the western or in the eastern part of the sound system, up near the barrier islands over time. Also, southern part of the Pamlico Sound, things are decreasing. If you look at the acreage that's lost, and also the loss in the central zone over time. And then here, if we look at Bogue Sound, the changes here are somewhat smaller, and that's because this is a big tidal system as opposed to Pamlico Sound. So the impact of that organic matter loading and other factors that decrease transparency are less than you would see in the sound itself. Now, I've given you a lot of negative stuff, and I wanted to kind of finish this talk by putting at least something positive in here. And that is that there are examples of where nutrient management has been able to restore SAVs. And of course, the Tampa Bay example is, people use that a lot, but it worked very well with decreases in nitrogen loading in this system that decreased the blooms, increased the transparency. So after that event occurred, the seagrasses were able to reestablish themselves. And, if we can get increased transparency back, or at least get in the upswing with transparency in Pamlico Sound by reducing nutrient inputs, we'll hopefully follow the same track. Chesapeake Bay, the summertime dead zone is decreasing in size, also attributed to decreases in nitrogen loading largely, and restoring hydrology is key to reducing this non-point source pollution action, obviously. And that will foster more resiliency in the face of sea level rise and storms. Now, I realize that this is not so easy, particularly with major events that just flood out the entire area that we're concerned with, but certainly enhancing riparian buffers, stormwater retention systems, having native vegetation wherever we can have it as opposed to barren soils, and of course reducing nitrogen inputs from fertilizers will all help in that direction. So there is some good news. We can get back to a more healthy and more sustainable system, but it's going to take a concerted effort in terms of nutrient reductions and adapting our land-based management to deal with these large storm events, particularly with sediment inputs to our systems. And the one thing I want to stress, and I always stress this to managers, even in China, that are in a hurry to restore their systems, these systems take time to recover. It doesn't just happen overnight. And if we are getting this increase in frequency of storm events, we may be facing a situation where the patient hasn't been recovered yet and is being impacted by some kind of new thing. It's almost like the COVID, new wave of COVID after the next one being too close together to allow for full recovery. So this is one of the things we're really trying to understand using ModMon and FairyMon, is what is the real recovery time that we need, and how is that being impacted by an increase in the frequency of these storm events. So lastly, here's sort of my summary slide. 40 years of bioassays has shown that nitrogen is the most limiting nutrient, but we shouldn't let phosphorus off the hook. N limitation has persisted in the open sound system. That is largely due to all that legacy phosphorus that's sitting in there. We don't want to increase the legacy phosphorus, so we need to really maintain phosphorus input constraints as well. And while inorganic nitrogen loading has decreased, organic nitrogen has increased. And this is really the topic of research that's being conducted by us and others. Further nitrogen inputs are needed while holding the line of phosphorus as well. We need to identify the inputs, particularly the organic nitrogen inputs, see where they come from, what we can do about reducing them or controlling them so that we can get below these bloom thresholds. And the new normal in high rainfall and tropical cyclones is reality. This is not just a fluke. We have long-term data sets that can verify that. And it turns out that other places are seeing the same thing, including Eastern Asia, the Chinese coastal regions, Japan, Korea. So climate change is, it's a real deal thing that needs to be taken into consideration because they strongly affect this relationship between increased nutrient inputs and productivity in the system. And hopefully I didn't go too far over my allotted time. I just wanted to thank the support that we've gotten, starting with NSF, of course, the state's environmental quality agency, the North Carolina Policy Collaboratory, which is actually administering the funds for ModMon and Ferrymon, the Lower New Basin Association, which has been a trustee partner and has also contributed to the effort, particularly with data management and putting things into graphics that some of you get every time Nathan Hall emails it. Sea Grant and WRI, and then the National Wildlife Federation, which supported ModMon over the past several years as well. Thank you, and I'm very willing to answer any questions. And lastly, I'm going to send some publications to folks at WRI so that anyone who wants to look at relevant publications on this can get them through the WRI office. Thank you.
And thank you, Dr. Paerl. That was awesome. No, you're right on time. You were actually right on the time schedule we discussed. It was great. We will post the publications on the NCWRA website too, so people can get access to them that way. We had one question already come in from Kevin Nunnery. Hey, Kevin. He said, Kevin was curious as to how nitrogen sources are determined, CAFO versus ag, and can organic, and it's a two-part question, can organic carbon be sourced accordingly?
Yeah, so this is work that we are conducting with Chris Osborne, or I should really say Chris Osborne is conducting with us, because he's really doing the analytical part of this using fluorescent signal for different organic sources, including urban stormwater runoff, animal waste, septic systems, and let's see what else, and natural sources, like coming from soils and forests, et cetera. So we are able to delineate the pie, so to speak, better using these fluorescent signals. It's called FluorMod. If anyone's interested in understanding how this works, I can also include the paper that Chris and I and others published on this. It's a program that basically looks at fluorescence emission versus excitation from these different sources, and then they give different patterns basically, and we can interpret the patterns nicely, even with the individual sources coming in together into the system. This is one big advantage over using, say, stable isotopes, which are good, but if you mix sources and you're trying to monitor those at the entrance of the estuary, you kind of get a mixed bag, and then you have to go back to looking at the sources anyway. So the fluorescence approach is a real advantage. We can also use it in bioassays, so we can actually see what's disappearing and what's staying around. And this is our big hope with regard to the kind of reactivity and linking it to what the algae are using, particularly the harmful algae.
Okay, excellent. Thank you. I had a question. All right, something that I observed, or I think I observed as you went through the long-term graphs from both the Chowan and the ModMon data showing long-term nutrient and chlorophyll A trends.
Right.
And you were talking about chlorophyll A on an overall increase, and you can see that in the back half of the graph pretty clearly, but it actually even looked like it was declining down to about early 2000s, 2001, 2002, and then takes off again.
Yeah.
And I was just wondering what observations you might have with regard to that pattern. Was that we didn't have any tropical storms in those years?
Yeah, here's the data for the Chowan. Okay.
Yeah, that's exactly what I was talking about.
Yeah. So what's happening is that organic nitrogen is starting to dominate the pattern in this, in this, well, in this graph. Also, we see it in the news. And I don't have the data from Pamlico River, but I have seen some inklings of data through ECU's program, and it looks like they're also seeing an increase in its organic nitrogen. So, you know, early on in the data set, we recognized we had a problem, right, with nitrogen. So here's the nitrogen reduction as nitrate, and that's great. You know, we're doing, you know, we're spot on there and reducing it. But look at organic nitrogen, it's increasing. And we think that that's due to changing land use, but also storms and more extreme events that are leading to these big flushing events that we see in our system.
Okay. Here's
And we
another.
think you know, we think there's a relationship between that organic nitrogen input. It's not, you know, there's some reactivity of that nitrogen. It's being used because there is a clear relationship there between, as you can see here, between TKN and the and chlorophyll A. It's, you know, it's, I think a lot of it depends on where the sources are, what they are, how reactive they are, and hopefully the work that we're doing with Renzo Zhao, who is at AMT, on the landfill leachate is going to give us a pretty good, interesting picture there, because he's able to distinguish those sources quite nicely using mass spectrometry. So that's really, you know, top of the line kind of stuff. And then we can do the response using our bioassay approaches to what he, you know, what he sees is coming out of those sources. And that's really what needs to happen. We need to be able to distinguish individual sources and then see how bioreactive they are. We've been doing some of that with animal waste. Chicken waste in particular is very reactive in terms of organic nitrogen coming from it. So, you know, I guess you really stay tuned on that. But that's an important question because obviously it's going up. I mean, this is the states. This is DEQ data, by the way, from the Chowan.
Here's another question that came in from the chat. In North Carolina, water quality, the water quality storm is a 1 inch or 1.5 inch for stormwater treatment. Are we now seeing that 25 to 100-year storms and the like could actually be having a higher impact on water quality with 1 to 2 events per year happening?
Yeah, I think the answer is yes, particularly if you go back to our Modmon data showing the loading that's going on for both nitrogen, phosphorus, and organic nitrogen. So I think I'm not sure what these. Yeah, I'm no longer sure what a 500-year event means because we've now had like 3 or 4 500-year events in the last 20 years. So somebody needs to explain that to me, how that works. I think we may need to revise all that.
You know, Bill Hunt has a really good talk. I don't know if the stormwater professor at NC State has a really good talk about how the recurrence intervals and the way we're looking at them needs to be completely revamped again. You know, that our current, like you said, the current idea of a 500-year storm is bordering on a useless designation at this point. Anyway, we got another question. That was Daniel Wiebke's question that just came in. Thanks, Daniel. Here we got another one from Frank Lopez. Frank says, because the watershed start in the Piedmont, is there evidence that sediment transport acts as a limiting factor in chlorophyll production?
You mean because of turbidity?
Turbidity and shading, I would assume. Not
for the algae, because it will for the seagrasses because they're stuck. They're on the bottom. And so, yeah, if you want to talk seagrasses, that's the big problem. You want to talk algae, not so much, because a lot of the bloom-forming algae, they can form surface blooms and get around that issue. So, and, you know, I'm sure that's an adaptive feature that the algae have, that, you know, if they become light limited, they bloom near the surface. The unfortunate thing about seagrasses is that they're stuck down there. So this is what's driving the system more into a planktonic dominated, you know, kind of algal bloom driven system versus what we would call more healthy, clear, macrophyte dominated system.
Okay. Yes, and Gene Robbins from USGS just put a link in there for a publication about the recurrence intervals and the like. Thanks, Gene. Had shout out to Gene.
Yeah, I'd like to get a copy of that. So
yeah, no, it's you can pull the link right from the chat.
okay.
Are there any other questions? I'll issue a last call for questions here. And seeing none, I will toss it back to Daniel. Hey, one last thing though. I know Daniel mentioned before we get started with this, nominations are out there, you know, please nominate people, nominate yourself. Absolutely. That's fair game. And volunteer for committees because NCWRA is a fully volunteer organization and it doesn't happen without the participation of folks on our board and in our committees.
So I will, before you kick me out or off, I will be sending these publications to Lee, and then
That works.
Yeah, and then she can communicate them to anyone who's interested. And there's going to be about, I don't know, 5 or 6 of them or so. Pretty easy to read stuff.
Excellent. We'll make those available on the website on ours and maybe WRI to boot. Thank you again, Dr. Paerl. That was really great. That was really what we had in mind in terms of what's going on out there in the current status.
Okay,
I appreciate it.
Stay tuned. You know, the Albemarle Sound is the system that you really need to keep your eyes on because it is brackish, susceptible to these blooms. And I think if you look at all the data sets that have been gathered through DEQ's program and ours and ECU and others, it has the clearest signal for eutrophication right now. So, you know, I just want to stress that Albemarle Sound is kind of, if I had to look into my crystal ball and say, you know, 15 years from now, what system has changed the most, that's probably it.
Okay, excellent. It's good to know. And we will be watching.
All right. All right.
Take it away. Thank you, Dr. Paerl. Take
Thanks,
it away,
Dr. Paerl.
Daniel.
That was really all I had. The announcements again. Remember, everyone, nominations are out through December 8th, and then we'll vote after that. And then again, I think Frank and my emails are in the chat. So if you have any desire to help with interest in the national conference here in 2023 and or volunteer for the boards, the committees that I named at the beginning, please message us. So with that, I appreciate everybody showing up, and we'll talk to you again at the beginning of next year.
Hey, Lee, can you send me your email?
Coming at you, Doctor.
All right. Thank you.
Thanks again, Dr. Paerl.
Take care.